started. Last talk of the workshop, right? Right. I made that mistake once before. XJ Wang, NYU. There's the title. Welcome.
The microphone's on. This is the last talk, so I propose that we give a round of applause to the organizers, Bruno, thank you for organizing this very exciting and stimulating workshop, and thank you for inviting me. So I thought I would like to share with you in this talk some of our work, ongoing work of the last few years, trying to go beyond the local circuit and think about how we can start to build a large-scale, multi-regional cortical circuit model. So some of you may know that for many years, research in my lab has been focused on some cortical areas that are especially important for cognitive computation, say in contrast to early sensory coding and processing. So we have been focusing on this particular area called the prefrontal cortex, indicated by blue across 6 different species, which is believed to be really important for many different kinds of cognitive processes. But our understanding is still very limited. In fact, as indicated by this graph, which is the number of papers published every 5 years with the keyword prefrontal cortex, you see that the interest and the research has going up only in last few decades. So it's relatively recent that neuroscientists really started to devote serious efforts to understand prefrontal cortex. So over the years, many people, of course, experimentally and in computation and neuroscience, trying to ask how we can really look at the cognitive functions, and different people tend to focus on particular cognitive process, as indicated here by given example names. Some physiologists working with monkey, a few working with rodents these days, say dedicating their efforts to, say, study simple decision making, to working memory, or to selective attention, valuation, inhibitor control of action, maybe a bit more abstract representation of behavior rule, etc. And in parallel, of course, we also try to develop some computational modeling to understand, each of those what we call building blocks of cognition. But at some point, I guess, as a field, we have this increasing awareness that each of these processes engages more than one brain region, right? So if you talk about the working memory in a delay task, the representation of working memory seem to involve many different brain areas, cortical, subcortical. And of course, in daily life, any flexible behavior depends on multiple of those building blocks. And again, each one engages a subnetwork of the brain system. So we really, I think, as a field, should start to think about how really we can confront this challenge and start to think about the bigger system going beyond the local circuit. Okay, so this is the motivation for the kind of research that we have been carrying out over the last few years. That's the question. So we decided to start with a recently published database about cortical to cortical connectivity in macaque cortex. This is a recently published work from Henry Kennedy's group, where they used a rather traditional retrograde tracer technique, but very quantitatively. They have a certain way to quantify the connection strengths from one area to another. So you end up with a connectivity matrix that's directed and weighted. Okay, so among the main observations are that whatever way they come up with to measure the strength of a connection, that weights those the values of that weight between areas span 5 orders of magnitude and can be fed with a log normal distribution. And the weight of connection between two areas is a exponentially decaying function of the distance between the two areas.
What is FLN?
So that's the way they quantify the connection weight, which has to do with the fraction of neurons that are labeled that project to a given target area from multiple source areas. So it's a relative measure of input weights to a given target area. Anyway, so we decided to use this database to constrain the structure connectivity in our dynamical large-scale brain circuit model. So we started with this subgraph for which we have this connectivity data with multiple visual areas, some prefrontal areas. And the experiment is being ongoing, so we continue to have more and more data for additional areas in the model. Now, to build a dynamic model, of course, the next question to ask is what would you do with, building dynamics in each local circuit? So our approach is to assume a very simple, so it depends on the questions, of course, that you want to investigate. You have to choose the right dynamical model, right? And to start with, we used a very simple threshold linear EI network for each cortical area. And that's actually coming from the literature from Martin Douglas group. And the model was kind of calibrated by the physiology of V1 in macaque cortex. So the idea is that we would use the same kind of local circuit model for the first round of simulation for each of the cortical areas in the large-scale model in the spirit of assuming there's a kind of canonical local circuit. However, we also assume that there are some quantitative changes from area to area, which would correspond to certain parameters in this equation that would be different from area to area. So for that, we look at a very interesting separate database about spine count that I'm going to show in the next slide, which seem to vary systematically along the cortical hierarchy. So this is an example from Elston showing the number of spines per neuron, which is different from area to area. Okay, if you plot the number of spines per neurons in this particular example, it will be the number of spines in the basal dendrite of layer 2-3 pyramidal cell. You see a systematical increase of that as function of the hierarchical position of the cortical area. Right? We will take this as a proxy about a proxy of the number of excitatory inputs per neuron that changes from area to area, and that we would incorporate that into the model, which would be this parameter, all right, HI, I being the index for the area, and that would increase systematically along the hierarchy. Right? So this is a rather simple model, but with some anatomical constraint about the area-to-area connectivity and this gradient of the amount of excitation per neuron, if you like, across this whole system. So this is a simulation of the model where you provide an input to V1 and watch the response from each of the cortical areas in the model. And by the way, this is the model really at the core of a PhD thesis by Rishi, who is here in the audience. Where is Rishi? Yeah, over there. Yeah. So he was the very brave and the risk-taking graduate student in the lab who really spearheaded this construction of the model. Anyway, so what you see is that different areas here are coded by different color. That the time course of response changes quite a bit from area to area, right? Another way to quantify, the time course of response to a given external stimulus is to, yeah.
All the areas have the same intrinsic time scale?
All the area has exactly the same equations, yes, the same taus, except again, the amount of excitation is scaled by that gradient, both for the local excitation and long-distance excitation. So this is another way to look at the time scale of dynamics in each local circuit. Here we drove, Rishi drove the system by providing a noisy input to V1. Okay, and you kind of see that the fluctuation has different time scales in different areas, right? Of course, you can quantify that by looking at the autocorrelation function of the time series from each area. Again, each one is given here in different color, and you can fit that autocorrelation function with, say, an exponential decaying function or a sum of two exponentials. And you can extract a weighted time constant that way. Then you find that indeed there's a broad range of time constants. Like early sensory areas, such as V1, has a time constant of about 20 milliseconds, consistent with the observations by people like Massimo Scaziani, whereas some other areas have time constants all the way up to a couple of seconds in this example. The x-axis is the hierarchical position of cortical areas, right? So I want to make a couple of comments here. Number one, in a way, this hierarchy of time constants is really functionally very desirable, right? You want early sensory areas to operate very fast, whereas some higher areas may be involved in time integration of information during decision-making process. You probably are familiar with drift diffusion model as a very simple model for accumulation of information in decision-making, and that assumes that the effective time constant of the system is very long, right? So you see here that a range of time constants somehow emerges from the system. Yeah.
So forgive me if you already said this, but is this a purely feed-forward model, or is
No,
there
no, no.
any feedback?
No, so that's a very important point to say it explicitly. I did not say it explicitly. So the connectivity between areas is given by the measured mesoscopic connectivity matrix from Henry Kennedy. There's a lot of recurrent connections. Among all possible connections, about 65% of connections exist. So it's a pretty dense graph or subgraph, if you like. But again, the weights of connections are very heterogeneous, right? Spanning 5 order of magnitude. You had a question, Ken?
Yeah, so this autocorrelation is computed from giving input to V1 or giving input to the area in question?
To V1.
To V1.
Just noise inputs, yeah. Yeah.
So that means that higher in the hierarchy, in fact, it's a cumulative effect of all the areas.
Well, it is, but that does not explain what you see. I guess, but it's a legitimate question, right? So you can have this, let's say, integration of integration of integration, right? That could give you a slower, more sluggish response. We actually did a model to see how much, right, that effect can account for the observed, dynamics. It's not enough for sure. But the other thing is that if you like, think about this is more about the decay time constant rather than the latency of response, of latency of onset response. It's more like how slow the response will decay away when the input goes away, right? Maybe it's also worth mentioning that the time constant is not a monotonic function of the hierarchical position, right? And that in part is because of this complicated feedback connection loops. So for example, area 8, which is frontal eye field, is actually relatively low in the hierarchy, light purple here. But it has pretty long time constant because it's really part of the frontal lobe. It interacts quite a bit with the other prefrontal areas higher in the hierarchy. If you randomly scramble the long-distance connection weights while preserving exactly the same set of weights, this is pretty much gone. Okay. So I know this is in a way it's really a mathematical or computational workshop. So I would like to emphasize that this is a very interesting mathematical problem that we are pursuing to understand better. What's shown here? Think about this as a purely linear system, right? Threshold linear. Let's say all above threshold, you have really a linear system. Then the time constants are just given by the real parts of the eigenvalues, right, of the system. So for each eigenmode, you can also compute the eigenvector, right? So this hierarchy of time constant should be understood in terms of how much each of the eigenmode or eigenvector involves a small part or big part of the system, and whether the small part or big part of the system are spatially together or not, right? So that's quantified here by those two numbers. So basically the x-axis, so each dot is eigenmode. The color corresponds to the time constant here. Blue is short, orange or red is long. For each eigenmode, you can ask what's the fraction of the nodes or areas are involved, right? Namely, the eigenvector is not, the components of eigenvector are not negligible for certain areas, okay? So if you go this way, you have more localization. Namely, you have less fraction of areas engaged in that for that particular mode. Whereas along the y-axis, that's a measure of how spatially together are the engaged nodes, right? So the higher you go, the more spatially localized those nodes are. They are together rather than scattered, right? You see, actually, so those are a few examples. Especially if you look at this particular one and that particular one, right? They have the same fraction of nodes, more or less, right? That are involved in each of the two, but the value along the y-axis are quite different. Then you see clearly here, right? For this one, the nodes that are involved are together spatially, whereas those ones are more scattered in space. All right. And look at this one. This is pretty amazing. You have eigenmode, right? That's really, really localized and with a pretty long time constant, right? How can you have a slow mode that's spatially so localized? Anyway, more generally, right? If you think this connectivity matrix from heterogeneity is an interesting one for macaque cortex, you can ask, what's the class of brain circuits and dynamics that give rise to this hierarchy of time constants? I think that's a very general mathematical question. Yeah.
So I think we discussed this before, but Nuoli and Carl Svoboda showed in the mouse that the frontal area, ALM, when you silence the thalamic input, it decays in 10 milliseconds. The activity decays in 10 milliseconds. Can that be accommodated with what your model says?
Well, okay, so there are several things about Carl Svoboda's finding. So one, of course, is that this is data based on macaque connectivity data, based on macaque connectivity analysis, and it's not mice, but also it's only cortex. In Carl's experiment, that's a delay task. And they found that the preparatory activity during the delay period is actually, I would say, not about representation of sensory working memory. It's about preparation for the motor response. And that activity depends on thalamocortical loop. So if you inactivate a thalamus, that activity essentially is gone rather quickly. It's not directly relatable. The other thing,
you were talking about a decay time constant.
Well, so you see, when they inactivated thalamus, the activity in ALM essentially disappears. What that means to me is that those areas, if that's true, right? Neurons in those areas, in that area in ALM, essentially don't get a lot of recurrent cortical inputs, right? It's as if neurons were independent from each other. They didn't talk to other neurons, which seemed a bit hard to explain, right? Given we know about the connectivity in the cortex. So the other thing I was about to mention is that this is, if you like, still a linear analysis. What we're doing now is to look at a fully nonlinear version of the network model to look at distributed persistence activity. And I think that's a better framework to really compare with those kind of experiments. And of course, we also have plans to build a similar kind of, using similar kind of approach to build a model for mouse system. Yeah.
Actually, I'm looking at this. How do you know there's any reality to these groupings that you're showing here, or these collections of nodes? I mean, is there any way to kind of validate that the model isn't just spitting out kind of random black dots? I mean, have you looked at these areas and tried to look for evidence that these areas are activated together in some task, or how should we view this?
Yeah, so we spent some time thinking about this. I guess one idea would be, this is a modeling finding, right? But again, I think in general, this raises a set of interesting theoretical questions about hierarchy of time constants and localization of eigenmodes in rather, I think, interesting circuit because it's based on real connectivity matrix. But you ask, how do you test this experimentally? I guess one way to think about testing this is to look at resting state measurements with relatively high temporal resolution, right? I was about to show a slide. Actually, this is a slide about one way, whatever way you measure functional connectivity, which is really a measure of correlation between activities between pairs of areas in a resting state. And then we have some predictions about the signature, right, of this kind of hierarchy of time constants that you could observe in functional connectivity matrix. So let me go on, if I can, to tell you a few other things. I'd be happy to come back to some of this at the end of the talk. So one of the things maybe some of you noticed and some have not is that in this, let me go back. In this plot, there's a huge attenuation of signal, right? So the fine rate here is about 100 hertz. By the time you get to the prefrontal cortex, there's this interesting slow after discharges, but the amplitude of response is ridiculously small, right? So essentially, the signal attenuates when it tries to propagate across the system. And that is a well-known problem in computational neuroscience. And so that's under the name of, signal propagation problem. You've probably heard about this classic work back 99. And many people have worked on this. But so far, this kind of studies, computational studies of signal propagation usually use rather simple architecture. Typically, the connectivity is purely feed-forward. Each layer is identical. You use some kind of random connection with the same weights, right? And typically, people found that either the signal does die out, or the system may become unstable, right? But under some conditions, like shown here by Diesman and others, you could have rather robust propagation. Now, we thought we should revisit this issue because now we have this kind of more realistic connectivity matrix with a lot of feedback projections, with a lot of heterogeneity in connection weights. It's important to see whether we can deal with this signal propagation problem in this more realistic macaque cortical large-scale system. So feedbacks sometimes really cause for headache, as Ken Miller also kind of indicated, and shown here. So suppose that you drive V1 here and watch the response in both V1 and the higher area, like prefrontal cortex, in this case ACC. So either you get a huge attenuation, so this is log, along the y-axis about the fine rate. You see V1 has a decent response over time, but again, when you're at the top of the hierarchy, the signal is basically gone. Now, you can try to scale the absolute connection strength a little bit to try to boost the signal propagation, but then you would end up with this kind of instability, basically because of the feedback loops, right? So now the ACC signal increases which through feedbacks, through complicated pathways, V1 actually can come back, so to speak. So there's all this big feedback excitation, and basically the system becomes unstable, right? And so we were really inspired by Camille's work on local circuits. So they came up with this idea of so-called balanced amplification. Essentially, you can increase excitation to induce this transient boost of response, but also increase feedback inhibition to ensure stability of the network. So we tried that, and this is the control case with attenuation, right? A very small response near the end of the hierarchy. But when we generalized that idea to this multi-regional system from area to area, so you enhance excitation from one area to another, but also enhance local inhibition to counterbalance and ensure stability. Now the signal is much better by 100-fold, as perhaps better shown here. So this is the response amplitude for different areas along the hierarchy without this balanced amplification in green, and the purple is the case when we used Miller's trick, so to speak, for this large-scale system. We also tested the signal propagation with spiking neuron version of the model. So this is the first time we actually have thousands of spiking neurons per each area in this large-scale system and look at the signal propagation. We found a synchronized burst that propagates. We also found another regime, asynchronous activity that propagates across areas. And of course, people have found both regimes previously too in a simpler feed-forward network. One thing that's kind of interesting is that we noticed that the signal is faster and more robust across the ventral pathway. And we went back to the connectivity histogram from the experimental measurements and found that the connection weights along the ventral pathway tend to be stronger compared to the overall connection weight histogram. So this is just confirming that the enhanced or balanced amplification mechanism still works with spiking neural network. Now, let me mention one more thing that's a bit surprising to me. There was this idea of so-called ignition phenomena promoted by people like Standerhan as a signature of awareness or conscious report. So basically the idea is that if you say use a near threshold stimulus, sometimes you perceive it, sometimes you don't. Consciously, they have some evidence using EEG kind of measurements that when you don't perceive it, the activities seem to be mostly confined in the posterior area. But when you report that you have detected a near threshold stimulus, there's this kind of a global ignition type of the phenomena. We found that, in fact, different types of areas in our model respond to stimuli of different strengths differently. In particular, the higher areas like prefrontal subregions in red seem to have this threshold effect. Okay, so if you are below the threshold, the activity is confined in the posterior area, but if you're above, you have this kind of global distributed activity pattern. And that, in fact, this nonlinearity, the threshold, in fact, disappears when you take away the feedback connections in the model. We have like 3 minutes. Let me just briefly mention 2 other things, maybe 3 other things. Just to, I think, highlight the kind of new questions I think we can start to investigate, at least formulate and investigate in this kind of large-scale model. So one of the things is about the interactions between bottom-up and top-down, right? So if you're interested in predictive coding, how the kind of predictive signal from feedback projections would interact with the reality through the feed-forward signaling. Well, To start to build a platform to address that question, we decided to incorporate layers, okay? Because the feed-forward and feedback projections are layer dependent. So we, and so I have to skip some details in the interest of time. We kind of build this next generation of the model based on some information about a cross-layer and layer-dependent connectivity. And we also incorporated a bit more interesting dynamics in this model. So now we have a superficial layer and a deep layer in each local circuit. And the superficial layer has stochastic damped gamma oscillations, whereas deep layer has slower low beta or alpha fluctuations. And so we connected them, and just to, sorry for the rush, we tried to use our model to reproduce some monkey physiological observations to validate our model and reconstruct physiologically hierarchy using this approach. And so, so this is the monkey data results, and this is our model computational finding, which the two are rather consistent with each other. Just to say that we now, I think, are in a position to start to ask real serious computational questions with this platform. However, let me end with one more puzzle and one idea about maybe addressing this puzzle. Namely, as I was mentioning, the connectivity is very dense. Each area gets input from maybe 30 other areas, right? There's this really urge and need to think about a gating mechanism that route the information to the right place to the right time, right? There's no way for such a system to work if we don't have such a gating mechanism. So we exploited this disinhibitory motif mechanism for gating. I don't want to skip this. Let me take a half minute to explain this. I think it's very promising to look at this important problem. So as you know, there's a diversity of inhibitory neurons. And roughly speaking, there are three major types. Let's say the first type in black target the soma or initial segment of axon of P amino cells, and that's well positioned to control the spiking output of P amino cells. There's a second type in blue that controls the input flow to dendrites or P amino cells, whereas a third type in green preferentially target the blue cells, right? So depending on whether the blue cells are very active, in which case you would block the input to the dendrites, or the green cells are activated, then they can suppress the blue cells and let a gate open, so to speak, right? And each of the three has distinct chemical marker, and this one, for example, express PV, whereas this one may express somatostatin or carbendin. There's a very strong evidence for this theoretically proposed motif in recent experimental studies using genetic tools. So I'm going to skip the details of how we incorporated this disinhibited motif for gating, and that paper is published if you're interested. But I want to mention that actually the input controlling somatostatin interneurons and the output controlling PV interneurons do not have the same proportion from area to area. So what's shown here, this is the paper that just came out, where they counted the different kinds of GABAergic cells throughout the whole mouse brain. All right? What's shown here in black is the ratio, basically, is PV divided by PV times plus the somatostatin neuron count for different cortical areas in mice, monotonically. Okay, this is the highest, this is the lowest ratio. Over here, you have more PV cells than SST cells, namely in those areas, you have more output controlling interneurons than input controlling interneurons. Whereas over here, you have almost 4 times input controlling interneurons that express motostatin than output controlling interneurons. What's interesting, in addition, is that when you just arrange the areas by this number, early sensory areas automatically fall off here, whereas higher areas like prelimbic areas, association areas are at the bottom. Namely, that higher areas, have a lot more input controlling GABAergic cells, which may be functionally very interesting if you think about gating. So let me end with this slide suggesting that, one way to start to think about this complicated large-scale brain system, right? What does it mean to build a large-scale system? Can we expect to find any kind of general principle given the complexity and the messiness of everything that we just heard? Well, one way we're pursuing at least is this idea of macroscopical gradient, right? So you have some canonical organization that may apply to each of the local circuits. At the same time, certain circuit properties, such as spine count per neuron, such as input controlling versus output controlling inhibitor neurons, may systematically change along certain axes in the cortical circuit. And if you are from nonlinear dynamical systems world, you know that some quantitative changes can give a rise to qualitatively very different behavior, right? Through what's called bifurcation in mathematics. So we are pursuing this idea that, maybe it's said here in the next slide, right? How to reconcile, on one hand, this unitary view of cortex and the modularity of different cognitive functions is, exploring this idea that quantitative changes can give rise to qualitative different functions. Of course, plus plasticity. So let me end by thanking people who did the work and be happy to answer more questions. Yeah.
I'm kind of curious how so I was thinking about finding hierarchy in the mouse brain a lot lately. So I was wondering how you define the hierarchy. Like, was it based on the layer specificity, like basically feedforward, so feedback connections, avoid the layer 4 and that kind of stuff? Is that what you use for hierarchy index?
Yeah. So in macaque, hierarchy is defined exactly based on the layer dependence of projections. In mice, I guess some people have tried, but so far we don't know of a clear, well-defined hierarchy, right? The plot I showed you actually is not based on a defined hierarchy. Actually, we just arranged areas by this number. And just somehow, early sensory areas we know, at least right, primary sensory areas we know who they are, turned out to be around here, whereas association areas like prelimbic, infralimbic areas turned out to be at the bottom. We don't know what actually is the best way to define hierarchy in mouse.
But on the previous slide where you showed the Granger causality, that was from the macaque.
That's for the macaque. That's indeed based on physiology. It's a good question whether you could think about using similar kind of approach to mice. In fact, the same group who did the monkey experiment attempted the same thing with human. So they used MEG data, and because that particular analysis was based on physiological data rather than anatomic data, so they actually applied that approach to MEG data and claimed they could find a pretty well-defined hierarchy in human. And of course, for human cortex, we also don't have an anatomically defined hierarchy. So maybe somebody could try that with mouse. Yeah.
As I recall, you had a slide on cognition that included things like motor control, decision, and so on. It did not, as I recall, include language. At the end...
In that early slide,
yeah.
you mentioned it, yeah.
At the end, there's a comment by Fodor on faculty psychology from modularity of mind, making the point that faculty psychology is, for example, impressed by the difference between language and thought. So let's call the stuff you have earlier about decision, motor, and so on, thought.
Yeah, would you call...
Do you have a theory about the distinction between that and language?
Would you call language building blocks of cognition?
Excuse me?
So I guess we use the term building blocks, basic building blocks of cognition. Those are kind of relatively simple, elementary processes. Let's say, how do you make a simple decision? How do you hold some information in your mind in absence of external stimulus? How do you suppress, prepotent automatic response, called inhibitor control? So those are kind of relatively simple, elements of cognition that people can study rather well with single neurophysiology in behaving animals. So those are the things we tend to focus on, so we can try to relate our model with physiology and the behavior. Language definitely, of course, is very important for human intelligence, but it's probably at the next level. We probably have to think about more elementary, more fundamental processes such as sequence, such as grammar, right? But I think it's useful to think this way, right? So, for example, the hierarchy of time constants turned out to be well received by people who work with humans, because there's this person, Yuri Hansen at Princeton. He studies speech comprehension, right? So you understand words with very short time scale, right? A very short scale, and then you integrate words into a phrase, from phrases to sentences, from sentences to narrative. And he showed that actually this kind of time integration on different scales involve different segments of the cortex. So there's this idea of a hierarchy of time constants that allow you to do that, right? But we're not there. We don't work on language.
Yeah. About the ignition phenomena that stand the hand, can you show the slide again? So in the hand's case, what is the analogy in your case for for the stimuli that can be preserved?
Yeah. So you're spot on. So the analogy would be that you put an input right at threshold here, and your dynamics is stochastic, right? So in some trials, in some individual trials, the activity will be confined in the posterior part of the cortex, whereas in some other fraction of trials, you get this more global activity pattern, right?
What are the axes of this graph?
The x-axis, this is the input intensity.
Ah, okay.
So it's like changing the contrast of a stimulus, right? So you just adjust the contrast of the stimulus right at the threshold of detection.